So my name is Josh, and we have here Duncan, who is a third-year PhD student in microbiology at University of Copenhagen, so that's in Denmark. And Duncan looks at novel antibiotics from the nose microbiomes. So an environment in your nose, basically. He's using bioinformatics, and he'll run us through like some of his research and his journey to where he came to be. Over to you, Duncan.
Cool. Let me just share the screen. Sharing. Cool. So today I'll be talking about the nasal microbiome as a novel source of antimicrobial. So I guess research insider number 15. You can follow me on at Duncan_Ng, and you can read more about it on my website, duncan.science. And talk more about this later on, or you can ask me questions, but my PhD is funded by the European Union Horizon 2020 program. So with that, I'll just move in. I'll go through the overview of what my talk will be about. So first, I'll talk to you about who am I. Then after that, I'll talk to you about antibiotics in general. Then about Staphylococcus aureus and why we're interested in it. Antibiotic resistance. Then followed by antibiotic discovery, a numbers game. More on that later on. And then the human microbiome as a potential solution and studying the microbiome. And then just a take-home message if it kind of went too much and you just don't remember anything. Just a quick summary of everything. So who am I? Well, I'm Duncan, obviously. I'm from Kuching, Sarawak. And for those of you who don't know where it is, it's that point over there. And I've had some West Malaysians who have no idea where it is. So that's an important note for you. We are known for our cats because Kuching means cats. So, and it changes according to the season. And now because of COVID-19, it has a mask on. And it's also known for the rosacea, snake plant, and orangutan. And these are both taken by me when I visited some of the tourist attractions. And next, my journey, I started off at A levels at Taylor's College, Subang. Then after that, I did a bachelor's in virology. And then after that, I kind of went into bioinformatics because I liked the idea of applying dry lab to wet lab stuff at the University of Glasgow. And that's me doing graduation. And that's a kilt, if you have no idea what it is. And then that's a picture of I took when I went to the Isle of Skye. Just like this island outside in the Highlands. So moving on, right now I'm doing a PhD in microbiology at the University of Copenhagen or Staphylococcus Serum Institute. And just to give an overview, Staphylococcus Serum Institute is a public health institute based in Copenhagen, things national surveillance. And I'm based there, but PhD students need to be registered at a university, so I'm registered at the University of Copenhagen, but I'm not actually based there. I'm also part of an EU project called Combating Antibiotic Resistance Training Network, or CARTNet, and you can kind of see the logo up here. And it's essentially, it consists of 13 PhD students, and we are scattered around Europe. We do different things. Some people are focused on drug design. Some people, like me and a few, are looking at novel antimicrobial antibiotics in either human, animal, from the soil. Some people are looking into phage therapy and all that. And these are some of the pictures I've taken in Copenhagen over the season. And here you have like during winter time. Here you have during spring. And here, anytime really. It's just one of the cool streets with colors. So moving on, then I'll start going into antibiotics. Oh, I forgot. We should do the poll. Yeah, it's just two very simple questions. The first one is just how many new classes for antibiotics have been discovered in the past 25 years introduced into the market. And then question number 2 is how long did it take for penicillin resistance to appear after doctors start prescribing it? All right, that's a lot of interesting answers. And we have 14 out of 18, 15. And when do we end the poll?
whenever. I guess there's not going to be more response.
Yeah, I think, yes. Okay. So that's all of us. Share results. So from the results, about 13% said 0, and the answer is actually 0. There has been no new class of antibiotics introduced in the market in the last 25 years. And there's a lot of people who said 4 to 10. And how long did anti-penicillin resistance appear after doctors start prescribing it? The answer is 8 years. It's not even, yeah. So it's quite a dire situation. So I'll talk more about this. Right, so now it comes down to the question of what are antibiotics? So anti means, again, biotic means living organism. And both combined, you have antibiotics. Generally speaking, antibiotic refers to the drugs that kill the bacteria, and antimicrobial refers to generally it kills the bacteria, but it might not be a drug. Like it can be bleach, and you shouldn't use bleach as an antibiotic because that kills people. Anyway, moving on, then we talk about the medical field and how it was revolutionized by this guy called Alexander Fleming, who was Scottish, and he discovered penicillin in 1928. And this is him, and he won the Nobel Prize in Physiology or Medicine in 1945 because of this. Next, I'm going to talk about penicillin timeline and how it is. So this guy, Fleming, he went on holiday and he came back and then he saw something like this, what you see here. Essentially what it is he has a lot of lawn assay of Staph aureus, which is just a bacteria, which I'll talk about later on. And he kind of noticed that there are some sort of fungal contamination growing on it and killing all his Staph aureus. And he starts wondering, oh, why are they killing them? Like, normal people, under normal circumstances, when you have contamination, you just throw it out. And for him, in this case, he saw and thought, that's interesting. They should look into that. And he did. And then that's how he discovered penicillin. But there comes the issue that Fleming couldn't purify the compound from this fungal he called penicillin notatum. So being a scientist and having to move on with his research, he passed this on to Ernest Chain, and together with Howard Florey and Norman Heatley, optimized penicillin extraction. So these people were like fungal people, and they know how to work with that, and they somehow managed to optimize it and extract the compound. But then upscaling was an issue. So they managed to extract the compound, but it takes 2,000 liters of penicillin notatum culture to make enough dose for one patient. So you can imagine all those bottles just to treat one person. The first person that managed to treat was this police officer, if I remember correctly. And when they started giving him treatment, he was doing really well, and he was actually like recovering. But then they ran out of dose because they couldn't produce enough, and then he eventually succumbed to his infection and, well, died. So that comes to the problem that even if you did discover something novel, if you can't upscale it, it's not going to be helpful for anyone. So then comes, they have this thing that is great, but they couldn't produce a lot of it. So, and around this time, there is, I guess, before that worldwide hunt, this was around World War II. So when World War II happened, a lot of the resources had to be diverted towards the war effort. So what they did, Ernest Chain and his two colleagues, he took the samples and he sent it to pharmaceutical companies in the US to hopefully try and produce more penicillin and find better penicillin. And that's where it comes to the worldwide hunt for a better penicillin producer. They need to find a way to produce more of it, and they know that the strain they have was not good enough. And as it comes, luckily, it comes in a cantaloupe. So this lab assistant called Mary Hunt went to a market, and this is when newspapers were advertising for, if you find any interesting mold, bring it in. So she went to a market and saw this cantaloupe, and it was covered in this golden mold. And she thought, oh, that looks interesting. So she bought it and brought it into the lab, which later found out that it was identified as penicillin genome. And it produced 200 times more penicillin than penicillin notatum, which in terms of revolutionized how they could produce it. And with that, they further enhanced it with UV light to some random mutations here and there, and as a result of that, they could produce like 1,000 times more. And with that, penicillin was able to be mass produced and distributed before, I think was it D-Day, so that when soldiers came in with any sort of cuts and infection, they could actually treat it. And it was a minor miracle through this combination of sheer chance that they could actually upscale it and save lives. So that's penicillin and how it got brought back to the market. But it's also worth noting that this is not the first time that antimicrobials have been used. It's been used since ancient times. It's just people don't really know it was antimicrobial. So in ancient Egyptians and ancient Greeks, they used moldy bread for their wounds. And we know mold in general produce quite a lot of antimicrobial substance to help them grow. And then even back then, they didn't really know what exactly it was. They knew how to apply this and prevent infection. There's also two examples more of this, which I'll talk about. So another one is this study that came out like quite recently. So they took a 1,000-year-old remedy, and this was a remedy combined with onion, garlic, wine, and biosalt. And the combination of these four things, they could make this, I guess, this salve, and they can put it on your eye, although I don't recommend you doing that on your eye. And then they found that that can treat infected eye, and it's published in this paper. And what was quite interesting was that each ingredient by itself couldn't kill the bacteria, but the combination of them together could. Then next we have this thing, which is also quite an interesting one. This happens in Northern Ireland. So essentially it's an ancient folk remedy. Back then, sorry, not back then. There's this part of Ireland, because of how the formation of the island, one part of it actually has a different soil composition. And it's been known for quite a while that this weird composition can actually help in ailments. So like if you have a cut, if you have like a toothache, they will take a bit of this soil, put it in cloth, and then put it on your teeth or put it on your wound, and then that would somehow cure them. And as they found out, the researchers went there and just like dig up some of this soil, just look into it. And they found that it actually contains like this Streptomyces bacteria, a novel one. And Streptomyces, if you are new to the antibiotic world, is that they are very good at producing antimicrobial compounds. And what they found is that they contain this novel compound. They could actually kill bacteria. So in that sense, the Tristan was actually effective at killing bacteria. People just didn't know that it was. They just thought, I guess, that it could. So that's something worth noting, that even back then, there are remedies that could potentially be antimicrobial. And there's like a whole field of this, I guess, archaeological search to find if there's weird combinations that can actually give you stuff. This is not to say that everything is working, but if it's validated by science, then yes, go for it. So with this, I'll turn the attention to Staphylococcus aureus and the bacteria that is a lot of the project is revolved around. So what is it? So Staphylo, meaning a bunch of grapes in ancient Greek, and coccus means for spherical bacteria or grains, and together they call it Staphylococcus. And this is what it looks like with an electron microscope. And this is exactly why it was discovered as a bunch of grapes that looks like circular, because this is what it looks like. So in 1881, it was first discovered by Alexander Oxton in pus from wounds. So he was a doctor, a surgeon, and he found like there were pus in the wounds. He took it, he put it on a microscope, and then he saw this, and which is why he called it that. And it's worth noting that Staph aureus infection were fatal pre-antibiotic. So before that, if you get it, you just die. Like there is no way around it. And the thing with Staph aureus is that it doesn't just cause one main thing, it causes like a range of disease. So you can get like minor skin infection, life-threatening condition, pneumonia, sepsis, meningitis, you name it, it probably does that thing. Now, it's 1 in 5 of the most common hospital-acquired infection, and it's mainly one of the main causes problem is a post-surgery infection. So if a surgeon cut you up, do something to you, and then close you back up, chances are they'll pump you full of antibiotics because they're worried that Staph aureus might be growing in your wound. And it's estimated that around 500,000 patients a year get it or is infected by it, and around 50,000 in the US alone dies from it. So you can imagine that it's quite a problem. And then just a short meme that's just time does not heal all wounds. So yeah, infection, bad. And generally speaking, you're like, oh yeah, antibiotics, right? Do you have antibiotics to kill them all? What's the problem? Well, the problem comes with antibiotic resistance. So what is that? Well, antibiotic resistance occurs when an antibiotic isn't effective at killing the target. So winding back, Fleming, the one who discovered penicillin, could see resistance in his culture. This was before it was even mass-produced and all that. He saw and noticed that resistance can arise if there is insufficient dose or given or for the right length of time. So if you don't give a therapeutic level to kill it off, or you don't take it long enough, then resistance can arise. And how does this happen? There's a quick lesson. So essentially, you have this here, lots of bacteria. Some of them are resistant naturally, and this is because of evolution and stuff. Like some of them do carry some variation, some have resistance to genes. When you take antibiotics, it kills off everything. And if you don't take enough or the right amount of time, some of them that survive can kind of grow in antibiotic just a bit. And then with that, it adapts and it can spread. And then once it starts taking over again, suddenly all the new ones are all resistant. Then I'm not going to talk too much about this, but just know that some of this drug resistance, it can be passed on to other bacteria that has never encountered these drugs, because that's just how the bacteria works. Genes get passed around. Now, with this in mind, I'll take you through what happened with penicillin when it was introduced. So penicillin was introduced in 1928, or well, it was discovered in 1928, but it was later on used as a treatment in around 45 or somewhere like that. So we have here penicillin, then after that treatment with Staph aureus. Because Staph aureus is such a problem back in the days that when penicillin was introduced, everyone thought, very good drug, use it on everything. You have any ailment, use it. Which then was a problem because like 10 years later, suddenly Staph aureus becomes resistant to penicillin. And then a few years later, it popped up. Then there was a pandemic where literally a lot of people have it, and then they couldn't treat it because everyone was resistant. So around this time is when the EU band together and then researchers and then introduced methicillin. And methicillin is just another antibiotic that targets the same way. And they're like, cool, we solved the problem, methicillin. Then 6 years later, they discovered methicillin-resistant Staph aureus. It was first found, and then a few more years later, there was a major outbreak. And then subsequent decades, more outbreaks of that. And then 10 more years later, it's everywhere. I think it's estimated that, at least in the US, that about 2% of people actually carry MRSA strains in their nose. Like, it was everywhere. So then they discovered, then obviously there was a race to find new ones, and then they found vancomycin as like the main treatment. And it currently, I believe, a variant of vancomycin is still the current treatment. Don't quote me on that. Then they also found, oh crap, suddenly vancomycin intermediate resistance was a thing. And then we found out that emergence of community associated. So what that means is that this MRSA is spreading within the community and actually causing a lot of disease and kills people. This is problematic. And then before this, before 2000, generally speaking, bacteria tend to be resistant towards one or the other. So if you use a combination of treatment, generally you would kill them all. So it was less of a concern because you knew that it could only be resistant to one or the other. And of course, the bacteria proved everyone wrong by doing both at the same time. And that was recorded. And then we fast forward towards now, you get, it's still not as big of an issue because there are still some drugs that can work on it. But at the rate it's going, it's only a matter of time before nothing works. So that is the timeline of the Staph aureus on drugs in itself. And next, I'm going to talk to you about antibiotic class. And I know we've mainly been talking about just a few types of antibiotics, but generally speaking, antibiotics break down to classes. And what classes just refer to is just that how they kill the bacteria. So in general, if you have multiple ways of killing the bacteria, you're more likely to kill the bacteria. So they can't evolve as quickly. If one doesn't work, you can use another type. And as you can see here, you introduce one, like 10, 15 years later, resistance appear. You introduce one, resistance appear. You introduce one, later down the line, resistance appear. You get the trend that every time a new class of antibiotic is introduced, resistance appear. And then what's the most troubling thing is that there haven't been a new class of antibiotic discovered in the last 30 years, which is a big problem when you think about it, because at the rate it's going, drug companies kind of improve what is already existing and they don't really make new ones. And it makes you wonder, why is it that they don't make more? So why are there new ones? First off, antibiotics are cheap. They are considered the cheap drug in the medical world. Compared to cancer drugs, they are like 1,000 times cheaper, which is why, and generally speaking, they're single treatment. Say if you get like an infection, doctors would prescribe you like, I guess, a few doses of it, but generally speaking, like a single treatment, and then that's you. But if you have cancer, generally speaking, you have to take treatment consistently for like a year to suppress or any infection, or if it was a HIV drug, it would, you would take it for a lifetime. So in that sense, because it's cheap, generally speaking, people only require a single treatment. It means that it's cheap, right? You also have the high failure rate. So only 1 in 30 new candidates actually make it through clinical trials, and it is very expensive to do clinical trials, so that is a really high rate. And that's the thing with new antibiotics, is that it needs to outcompete with the existing ones. So the existing ones are like really good. So if you are a doctor and there is a new antibiotic or a new drug, and you're like, why would I use or buy this drug when I know this one that has been prescribed for the last 20 years? Like, it's there, it's reliable. And that is a lot of the outcome for a lot of the, I guess, drug approval. It needs to be better than the existing ones. And needless to say, we've used a lot of the good ones. And also, antibiotic resistance equals wasted investment. So one of the things is that, like many things in the world, it's reliant on profit. So if resistance appear, it means that whatever money you've put into antibiotic resistance is just wasted. And also, if you find any new stuff, generally speaking, doctors tend to keep it as a last resort drug. They don't want to use it because if they use it, resistance will appear. So if they keep it sparingly, it will last longer. So I guess it boils down to money, that in general, that if investors don't see a point in antibiotic and it doesn't turn a profit, that people don't really want to look into it, even though it saves a lot more lives than, I guess, your typical cancer drug. With that, I will now talk about how antibiotic normally is discovered. So this is very generalized, and it's just to give an idea. So say it starts with getting a sample, like a soil with lots of bacteria in it. You try to isolate that bacteria, then you test it against target. And then what you're trying to do is get what Fleming saw initially, where you get like one bacteria and then kills the target. And then they essentially just do it a numbers game. This here is a Hamilton robot. And what it is you can test a lot of things very quickly. And you have entire companies with entire floors full of this, consistently testing stuff to see if it works. And if there is a hit, they'll take it and find out what kills the target, and then is it safe for people. And some of the problems with this last step, like generalized problem, is first, less than 1% of the soil bacteria can be grown in the lab. And this is not for any particular reason. It's just because the soil is a very complex environment, and it requires like some of these bacteria can only grow when there's like 5 other bacteria that produces a certain compound and it can grow, and so on. So even that, like imagine big, and suddenly you only have less than 1% to work with. Then even if you did find the bacteria that could potentially produce something interesting, you need to find the right conditions to grow it so that they actually express the compound, which is also a problem because some bacteria only produce antimicrobial compounds when they are stressed. But then what is stressful to them? It requires a lot of fine tuning and stuff. Next is find out what kills the target. But then it comes down to the question of, does it actually kill that with a compound or not? And I'll talk more about this about bacterial competition, but generally speaking, sometimes they don't kill them in the traditional sense of like, I produce a compound that targets you and then you just, well, wither and die. And is it safe for people? For example, bleach is a great antibiotic, but the side effects may include death. So, it's something to take into consideration. Like in general, people try to get antibiotics that are not toxic to mammalian cells. Like there are some antibiotics that actually target your central nervous system, which obviously people don't use because it kills people. So all the things need to be in consideration when you look for antibiotic discovery, which comes down to the question of the nasal microbiome. And what is it? Is probably the start of the question. And the microbiome just refers to all the organisms, virus, bacteria, fungi, protozoa, living in an organism environment that, they exist on your floor, your skin, your nose, your gut, everywhere. So the reason why you look into the nasal microbiome is that, so Staph aureus, the bacteria we're talking about, that was a big problem. About 1/3 of the population actually carry it in your nose. So like in the front bit of your nose, they estimate about 30 to 40% is the general consensus in the field. And there was this study that showed that twin studies that show that it's not genetic. So, why do some people carry it and why some people don't? This paper showed that it is not because of genetics. And then this paper further on explore that by looking into the microbiome, they saw that some bacteria are present, and when they are present, Staph aureus is absent. So this gives the implication that some bacteria are actively excluding Staph aureus from the nose, which opens up a lot of discussion on, ooh, that's interesting. It can do a lot of interesting stuff. So that's where my project kind of stems from, that you can kind of see this effect. So next, I'm going to talk about how this can happen. And this is in the competition of the nasal microbiome. So first thing, attention site. So if you can't stick, you're gone. And that's what is shown in this paper here with S. epidermidis, and it can produce this EST. And what it is, it prevents Staph aureus from, lack of a better term, sticking onto the nose epithelial cell. I guess it's epithelial. Yeah, epithelial cell. And if it can't stick, it can't grow, and actively excludes it. And then that's how it can grow well. That's yeah. The next one is nutrition. It starts your opponent to death by eating more efficiently, is how I would reduce the science. And it was found by this paper where pseudophores, or I would call it iron sponges, it's like this compound that's produced by bacteria, and it kind of pulls in iron from the environment and lets the bacteria reabsorb it into their body. And for those who are not really that into biology, who don't know their biology as much, iron is needed for a lot of enzymes. And if you don't have enough iron, you can't do the main processes needed to live. So by excluding iron, it's one of the strategies used by the body too. If you pull off enough iron from the environment, bacteria cannot live and they just die. So if you can outcompete Staph aureus with iron, you can starve them and essentially like grow better. So hence comes the statement, starve your opponent to death by eating more efficiently. And then the third thing is you can do antimicrobial. So at this rate, you literally shoot them through the heart, and because you're attacking the main essential part of them, and then kills them. And then they found this in luptanin, which can inhibit Staph aureus directly. And this is a bacteria that can also be found in the nose, and they show that it can produce this compound that actively inhibits Staph aureus. Then you can get host immune response. And what this means is that you get someone else to do your dirty work. And it's been shown in this paper where, so instead of making a compound that directly targets Staph aureus, what you do is that you get the body you stimulate the body's host immune response to then kill Staph aureus for you. Yeah, there's a lot of details to go into that, but just know that bacteria can also manipulate the host response to target, yeah, I guess, enemy. And those are like the few main concepts you need to remember about how bacteria can compete with one another. Bear in mind, this is not exclusive towards human. This can also be seen in pigs, or any other animals, or even plants. So this competition thing happens quite often. So how do we study the microbiome? There is two main methods. There are other methods, but I'll be focusing mainly on these two. So we have this amplicon sequencing, or 16S RNA, and you sequence a conserved chunk of DNA. And think of this bottom here. There is this circular thing with this one red bit here. These three represent bacteria. And this tiny little chunk is called amplicon sequencing, and they are conserved regions. So there are regions in the DNA where it is quite similar in all of them, all the bacteria, but there's slight variations in it that you can kind of tell what they are. So they're kind of similar to looking at a passport cover. So if you see this one bacteria, for example, you see this one region, it's like a person. You see someone with a passport, and you're like, oh, you have a passport from, let's say, Denmark. And you're like, oh, cool, you must be Danish then, living in Denmark, doing all the Danish things like cycling and all that. But in fact, one of the problems with this is that you only know that he's Danish, but you don't know if it's a guy or a girl or anything. You don't know what languages he speaks. He could be a French person for all you know, and you wouldn't know because all you have is like one tiny snapshot of it. So that's one of the problems with amplicon sequencing. So you can kind of tell something, but it's harder to infer. Like, for example, if you were to know that someone is, for example, Malaysian, then you'll probably know that that person is inclined to believe that nasi lemak did not come from Singapore, as in a case example. But of course, Anyway, it only sees one part. Then the next part you have is this thing called shotgun metagenomics. And I use a money flying away emoji because it is quite expensive. It's like 20 times the cost of an amplicon sequencing. And it can be more expensive depending how much you want to pay for it. It's one of those things. And it's kind of like Facebook stalking. You know more stuff, but there's also limitations. Like you can, learn more about this person, but that person might have a lot of profiles. So it's kind of hard to know if this person is actually what it is looking at, because there's more things to it. And there's also, with metagenomics, you sequence everything and then you kind of combine everything and hope that whatever you make from the mess is real, which sometimes you don't know if it is real or not. So those are the two main methods. And with that, I will talk to you on how I apply these things to my PhD with all the back knowledge that you know of now. So first things first, I have to get a lot of nose samples from healthy individuals. This was luckily done by someone else, mainly a doctor, because it needs to be done by a doctor, a clinician. So you get a lot of all these nasal samples, and then next you process them. So what you do is amplicon sequencing to identify what the bacteria and count them. One thing I forgot to mention is that amplicon sequencing, that one little bit is very conserved and it's only found in one bacteria. So if you manage to sequence a sample and you find like 20 of these genes, then it is quite accurately to say that you can say that of the 20 bacteria, 10 of them are bacteria A. Then you can roughly say that in this sample, 50% of the bacteria is bacteria A and 50% is bacteria B. There's more things to it, but generally speaking, that holds true. And next, after you've done this and you identify how many bacteria and how many counts there are in all of them, you can do a differential analysis. And what that is you have two groups. You compare group A, group B, and you see what the difference between group A and group B is. And in this case, it would be Staph aureus present, Staph aureus absent, and then from there you can get a potential list of candidates of like, you only see them in sample A. And it could be because they produce something, it could be because Staph aureus is killing them quite well, but generally speaking, that's the plan. Then after that, you get more data to throw in to try and understand Staph aureus carriage more, because there's a lot of, factors that can affect carriage, not only just the bacterial community. An example would be smoking. A few studies have suggested that smoking can reduce Staph aureus carriage, and so on. And they found that men actually carry more Staph aureus than women. That's also another thing that you have to factor in to this kind of stuff. So the third thing, after you get all this data and a lot of the samples, what you do is that you find samples that have like a lot of the candidates you think might be quite interesting. Then you go in the lab and isolate it. One of the bright side of working with nasal microbiome is that a lot of the bacteria you can actually grow without problems, unlike in the soil. Like a lot of the bacteria, it's less complicated, and you can actually see all the things. And then the fourth one is test if the bacteria is interesting. Like, you can isolate it, but then you have to test it, sequence it, compare it with other genomes. Is there anything interesting out there? There are a lot of software out there that can help you understand this better and see if whatever you find is novel or not. And with that, I will conclude with some take-home message. One, antibiotic resistance is a huge issue. And I know right now the focus is on COVID-19, but the next big medical challenge that all of us have been seeing coming is antibiotic resistance. And that's something we all need to, I guess, lack of better term, prepare for. It's quite similar to global warming, where it's hard to picture how it can impact your life until it does. And then when it does, it's quite bad. Second one is that the current model for antibiotic discovery is broken, and there's been like a few ways countries have been trying to maneuver this by introducing new system and all that. It's only a matter of time to know if it actually works or not. And three, the human microbiome has a lot of untapped potential. And again, I'm only focusing on the nasal, and I don't have enough time to dive into too much of the other stuff. But there was a few papers that they have discovered a novel antibiotic from the gut too. So it's not just in the nose. But generally speaking, if you know that whatever bacteria you're interested in exists in those niches, then you can hopefully find bacteria that normally targets them too. And with that, I would like to thank Mbios for hosting me, and I will take any questions. Thank you for listening.
Wow, nice talk and very well timed as well. So there's a few questions. I'll just throw them out at you as they come along. And anyone, if you've got more questions, feel free to add them, and we'll try to collate them and answer as many of them as possible. So the first one comes from Hui Shi, and she asks, how are antibiotic classes defined? Is it based on the mechanisms by which they destroy the bacteria, e.g. breaking the cell wall, or how are antibiotic classes defined?
Right. So don't quote me on this because I don't actually know too much about it. But from my understanding of what the classes mean is that they generally target a specific bit. So for example, penicillin would target like the peptidoglycan on like the cell wall, I guess, and disrupts that. And that is referred to as a class because it all targets one. And generally speaking, the reason why they group it into classes is that when one resistance arises in a certain class of one particular antibiotic, that can usually translate to another antibiotic, like of the same class. So because they're all working in roughly the same way, then that's how you get the different classes.
Okay. Yep. So the next question is there a reason why Staph aureus is good at obtaining resistance? So it does how, and I'd add to it and ask, is it any more different than other sorts of bacteria?
So the answer to that is actually yes. They are very, very good at adapting. So to give it a perspective, why Staph aureus is such a good grower. So in the nose, they can produce up to like 10 different kinds of like proteins to control their environment to grow better. That's one. Two is that they are known to have like this thing called an acecine-backed element. And what it is a genetic mobile element. And it allows them to transfer genes quite easily, like transfer from bacteria A to bacteria B to bacteria C. So if, say, a particular Staph aureus is resistant to something, then this mobile element can propagate into other Staph aureus, even without any sort of treatment. They can just like spread it. So it presents quite a problem because they can quite easily take in new genetic material, and some of this genetic material have resistant genes. And with that comes all this problem.
Okay. Yep. Thanks, Hakim, for that question. And next, we have Lei Hian's question, which is, does silver ion solve the bacteria-resistant problem? Silver ions.
Silver ions? Not, oh, like the use of silver or like, I guess, metals in general to kill bacteria. In general, yes, I guess. But I guess it also comes back to the problem of things that generally, so I think what the guy is referring to is, say, silver on a surface, I presume, rather than injecting into people and hope that it works. Yeah, I mean, I guess it kind of kills the bacteria, but the problem with a lot of these things is that it needs to be specific towards the bacteria. You don't want to kill the host while you administer the treatment, because that's kind of defeats the point of a treatment. So in theory, yes, it works, but a lot of drugs need to be very specific for human uses. Like it needs to be very targeted and doesn't affect the host.
Okay, cool. Yeah, you don't want to get metal poisoning out of it.
Yeah, exactly. And that's the problem with just metals in general that people don't tend to use.
The next question comes from Jiazheng, and it says, are there interactions between microbiome and host genetic factors?
So it's one of the, I guess, disputed topics, actually. It's yes and no. So some of the microbiome can actually influence the human body and the other way around too. Because when you think about it, I'm gonna just, don't quote me on this too, because this is just a rough idea on what I think can happen. So this pertaining to, again, talking in a field that I don't really know, the brain-gut axis. So one of the things that I vaguely know roughly about is that depending on what you eat, it can affect your gut, and what you eat also depends on what your mood is. So in some way, you can influence your own gut microbiome, and with that, the gut microbiome can also produce compounds that affect you as a human. So it's kind of both way, I guess. Yeah.
Okay. Yeah. So a follow-up question would be, so what are the biggest factors? I mean, you mentioned, say, nutrition, diet that influences this microbiome in the gut. Any other things other than genetics and nutrition that you can think of?
And off the top of my head, it can also be sometimes just a more invasive species. So we kind of see that in aureus in the nose. There's this particular clonal called CC398, and it's associated with MRSA in pigs. And what we see is that every time someone, say any one of us, go into a pig farm where this particular clonal complex is quite common in the pigs, they go into your nose and then they spread like wild. But you can suddenly see that it's just like, oh, there's none, and suddenly like a big spike in a lot of them. But over time, your nose microbiome tends to slowly come back down. And it's quite interesting to see like the transition of like spike and then back down, which suggests that some of the bacteria will eventually fight off this, I guess, more invasive species. And sometimes they fail too, and they just like persist. If that answers the question.
Yeah. So I'll jump into a technical question first. So I have no understanding of what this means, but
that's perfectly fine.
So Patron asks, what do you find most challenging analyzing both amplicon sequencing and shotgun metagenomics data? And so far, what have you learned from analyzing these nasal microbiomes?
So without going into too much detail, because there's a lot of things to talk about, one of the problems with microbiome study is that it's very dependent on how you swap the data. If you swap more data, if you swap samples, the more vigorous you swap, the more likely you are to get like a host DNA through, which means that you have less bacteria DNA to work with. And depending how deep you sequence, the more you sequence, the more likely you'll find stuff. So normalizing the data to compare it with each other is also a problem. how you extract the DNA is also a problem. If you extract it different ways, you can introduce biasness into your data. There's also the batch effect, depending on what you use to sequence. Then after that, there's also like various things like Illumina bleed over is a problem too. So what that is if you have sample A, sample B, sample C. Sample B has a lot of bacteria A, and sample A and C doesn't. Sometimes within the Illumina flow cell itself, this can bleed over, and then it causes some data, the sample A and C, even though they don't have a certain bacteria, to appear. So that a lot of these things need to be taken into consideration to hope that you get actual results. It's quite a disputed topic in the microbiome world, I think. And I guess for the metagenomics part, just very quickly, one of the problems is closely related species. So sometimes in the nasal microbiome, for example, you see a lot of Staph epidermidis, and it's just another strain of Staphylococcus. But Staph epidermidis, literature says that they exist as a polyclonal, so you get like different variations of it, but they are the same species. Now, if you sequence a sample with that, and you know that there's a lot of polyclonal, sometimes you can't split up the reads for like individual. They call this binning. If you can't split them up, and when you assemble them, what you get is like this weird hybrid that consists of like actually 3 or 4 bacteria into like this one genome. So with that, it also presents its own problem. Like, are you sure what you assembled the genome, is that actually what is present in the sample, or is that just a weird representative from like 5 different species? That's one of the, I guess, biggest challenges in metagenomics in general, on top of it being quite expensive and computationally very intensive.
Cool. I'll pretend I understand how it goes.
Yeah.
The next question comes from Jake. Lots of hype on antibiotics altering gut microbiome and human health. Anything known about if the nasal microbiome is shaped by our ongoing antibiotic usage and links to health conditions?
Yes. So one of the things so I'm not sure if you've noticed it, but when I do my studies, that we intentionally pick healthy individuals, and I might not have mentioned it, but we exclude them if they've used antibiotics. And generally, when you take antibiotics, you also notice that it's like a pill or like an injection. What happens is that antimicrobial antibiotic actually goes into everywhere in your body. Like it doesn't just go into your wound. It literally gets excreted everywhere in your body. And because of that, your entire microbiome can actually change just by you taking some antibiotics. And with that, you do see that some patients, when they take too much antibiotics, the one clear example I can think of is, I don't remember what disease is that. The one where it clears off the gut, and then it causes a buildup of C. diff. Yes, that's the one. Calcium deficit, I believe. And essentially, it's an opportunistic bacterial infection, and because of that, you have a lot of gut problems. And that's because of overuse of antibiotics. That's in the gut, and in the nose, you can see sometimes when you clear it, in the nose, Staph aureus tends to colonize too, because suddenly there's this whole empty house that a new bacteria can move in. So generally speaking, those that can grow significantly better tend to move in, which tends to be quite bad for the host.
Yeah. So I mean, this is one of my own questions, and it's some slightly off topic in a sense. But there's a lot of probiotics and things like that in the supermarket, and how effective are they in like doing whatever, introducing new things to your gut microbiome?
Yeah. Yes, I was fully expecting a question like this actually. So for the gut, there is a few papers out there that showed good, bad. One of the problems should highlight with any probiotic is that there is a lack of regulation with probiotics in general. People claim that a particular strain of, bacillus is good for you, and the market actually don't regulate that. So you don't actually know if it's actually good for you or not. there are quite a few papers out there that show that, yeah, there was this probiotic drink in, I believe, in Thailand. And they found that the bacillus probiotic drink actually helped alter the gut and prevent colonization of Staph aureus. It's a very long paper, and they did a lot of experiments. It's a Nature paper, that's why. And yes, to an extent, it can help, and it has quite a lot of C. diff thing with the transfer of the gut and all that. On the other side of it, there have been SEU studies too that showed that in ICU patients, they gave sort of probiotic to help them, I guess, get better. And what they found is that the probiotic pills with the bacteria in it is actually causing some of their infection. They actually found the same strain of probiotic bacteria in their blood. So they're like, oh, this is bad. And that's expected because you don't actually check the bacteria for resistance, or no, sorry, not resistance, virulence genes, the genes that cause disease, which in turn, sometimes some of them do have it and you just don't know and you take it, and obviously it causes disease, which is bad.
Okay. Yeah, I'll be more cautious when I take mine.
Yeah. Like when in doubt, don't take it. It's probably the safest option.
Okay. So Hakim has got a comment on your shirt actually. So off topic, does your shirt refer to genome sequencing?
So it's actually not. Funny enough, I got this shirt as a birthday present for like this channel called TLDR, or it just translates to don't too long, don't read. But I guess it can be applied. I didn't think about it that way, but yeah, I will.
Thanks, Hakim, for the punny comment. And the other thing is, I guess with what is unique about you from the rest of the people that have been featured on RIs is that I think you might be the first one who's doing a PhD in Denmark, Copenhagen. So I'm not sure if the audience is interested in how different the experience is in Denmark, because you've also did your undergrad and masters in the UK, in Scotland, and then you've changed to you found a PhD in Copenhagen. So how's it different? How's it like? Any?
So I guess I'll make a lot of the comparison with the UK system because I kind of know it better. So I guess also I should probably add a background on how I ended up here. Is that I started off when I finished my master's in bioinformatics. I thought, oh, I kind of like doing science. I'll just try for a PhD. And then I kind of applied everywhere in the world. Like I think I sent out like 20 applications or something in like various parts of the world. And then I got 5 interviews on like various places. One of it just happens to be in Copenhagen. And so when I applied for my PhD, is that because bioinformatics is such a flexible field, I didn't mind going into it. It's just to give you a perspective of like what the 5 came back as. Once in the interview, one was looking at ants and how their microbiome can affect their evolution. The second one was studying about metagenomics in soil in New Zealand, and just for surveillance and how that translates to pathogen. The third one is this one with Staph aureus and all that. The fourth one was a diagnostic company trying to find like a diagnostic way to determine something. I don't remember what. And the fifth one was trying to study how the gut-brain axis link together with the microbiome. And this place apparently has a germ-free pig model that you can go wild on. So in that sense, I applied to a lot of things. And the reason why I really like this one that I'm in right now is because it's actually funded by the EU. And what that means is that one of the requirements for EU, or at least PhDs under the Horizon 2020 program, is that their requirement for students is that that student must not be from the country, the host country, or have not lived there in the last 2 years. So what that means is that they encourage movement from like other parts of the world, which is why I ended up here. Which is, yeah, and overall the experience has been quite good. So compared in Denmark compared to the UK generally for a PhD, what you need is to write a very thick book that's 300 pages, then or something like that, and you have to defend your PhD in a room with your supervisor and two other persons, and for like, I don't know, 4 hours, 5 hours, something like that. Well, here, generally speaking, you have 3 years. You try to write up about 3 manuscript equivalents of material. Then after that, it doesn't have to be published. This is just the rough idea is that you write 3 manuscript worth of material. Then after that, you write a thesis around this 3. So you have like a story that goes with it. And then it's a public defense that anyone can come and visit. And then you have like 3 other people come in with your PI, and then you kind of talk about it, present, and so on. So overall, I prefer the system here because I don't need to write a 300-page book. And also the book is A5, and it's like, what, max 100 pages? So yeah, it's different, but it's actually quite nice here.
Okay. When you do that, I've seen like in Europe they do this public defense thing, which is kind of daunting in some ways. So can the audience ask questions?
Yes. So there is 2 parts of the defense. The first one is a presentation of your project. It's about an hour long, or less than that. And then the audience can then ask some questions. Then after that, it moves on to the actual PhD defense, where it's like another hour and a bit of you having to show additional data or justify some of your claims in the book or your thesis, and like why you said this and that, and how do you. So yes, audience can actually ask questions. And because of the age nowadays with COVID-19, a lot of this has been moved online, so even more people can actually watch these defenses.
Good luck with that. You're in the 3rd year, so it's not too far away for you in the next year.
Yeah, in theory, I'm hoping next year, somewhere in May, June, maybe. We'll see.
Yeah, good luck with it. Let us know, and when you have more findings, you can share them.
Yes.
From the questions and response, I'm pretty sure a lot of people are interested in your work. Yeah, cool. Any parting words from you?
Science is cool, but it's also very frustrating. I think that's something that people don't generally highlight enough of, is that anyone who's interested in science, the journey from where you are to, getting a major interesting discovery is that there is a lot of hardship that is not shown. And sometimes I'm sure some PhD students will agree with me and researchers that it's so much easier to just be a medical doctor sometimes, because all you need to do is just, like there is a test. And then once you're done with the test, you move on. But to get like the right kind of, I guess, collect enough data and do stuff, then yeah, it's hard. But then it's interesting because what you're doing, no one else has done before or have seen the results. So in that sense, it can be quite exciting and rewarding. But of course, it's hard work.
Yeah, it is. So, but very well put, right? It's
yes, exactly.
Pushing the frontiers of science. You're finding out things. You might be the first person in the world to know what's going on here with what you're investigating, and very well put. And with that, a round of applause to you. Yep. With that, we draw this to a close, and I like to just inform everybody. Next week we'll have Peichin Sabrina. She's joining us in the audience. Next week she'll be discussing plant epigenetics and bioinformatics. So also bioinformatics, but I'm guessing here now it's with plants, and we look forward to see you again next week. Right. See you, everybody.
Right.
Take care.
See you.
Bye-bye.